So I am Dr. Shiban, MBBS DCH, postgraduate from Madras Medical College, and this is our NEET UG MCQs, and this is our part 80. My first video itself is a repeat last year MCQ. This is last year MCQ. Biolistics is also called. Any idea? It is also called gene gun. Okay, that is other is called biolistics. It's called gene gun. This is a last year MCQ. Gold or tungsten is used in which? In your biology textbook. Where? Gold or tungsten is used in biolistics or gene gun. Repeated MCQ. Match the following point. Biolistics. Okay, what is the question? Gold or tungsten. Gene gun is used in plants only. True or false? Gene gun is used in plants only is a correct statement. Okay, these are all the MCQ points. Okay, so what do you mean by the word biolistics? What do you mean by the word? Any idea? Cells are bombarded with gold and tungsten in plants. That is called bombarded. Same word your textbook has given. Bombarded in plants. So it is called as biolistics. See, all the MCQs I have prepared from your NCERT 12th standard textbook only. Okay. What is the use of disarmed pathogen vector? Disarmed pathogen vector use. When it is allowed to infect the cell, what happens? This is the use of disarmed pathogenic vector. Okay, when it is allowed to infect the cell, disarmed. Identify and eliminating non-transformants. By what? Identifying and eliminating non-transformants is called selective or selectable marker. Both are same. Selectable marker. These are all boring MCQs, but rank deciding MCQs. Yes? Good. Selectivity permitting the growth of transformation. Very important MCQ. Very important. What is the answer? The answer is selective marker or selectable marker. You must know the question, not the answer. Question is important. What is the question? This is the question. This is another question. Answer is same. But the questions are different. So whenever they ask a question like this, you must answer selectable marker. And when they ask a question like this, you must answer selectable marker or selective marker. What do you mean by the word chloramphenicol? CHLOROM PHENI col. Chloramphenicol. What is it? This is a match the following MCQ. Chloramphenicol is an antibiotic. Chloramphenicol is an antibiotic. Can you tell me 4 antibiotics? Can you tell me 4 antibiotics? See, these are all 4 MCQs. Caniamycin, antibiotic. Tetracycline, antibiotic. Chloramphenicol, antibiotic. Ampicillin, antibiotic. Four MCQs are here for in front of you. 4 MCQs. The answer is antibiotic. Suddenly they will ask, tetracycline is a plant, true or false? You must know it's an antibiotic. Caniamycin is a flower. That time you should know it is an antibiotic. Not easy to remember. Read it thoroughly. Caniamycin, tetracycline, chloramphenicol and ampicillin are antibiotics. Made it very clear. So antibiotic resistance used where? Antibiotic resistance. Selectable marker. Okay, not selective. Selectable marker. The question is antibiotic resistance. When they give 4 choice, you cannot answer because they are dry MCQs, very dry. Antibiotic resistance is related to the word selectable marker. Selectable marker for which is the selectable marker for antibiotic resistance? Selectable marker for antibiotic resistance. Any idea? They will give you 4 organisms. You will get confused. Or which is true of E. coli, you must write selectable marker for antibiotic resistance. Piece of DNA. A piece of DNA introduced into the host bacterium is called mutation. True or false? It is called transformation. Transformation. Piece of DNA introduced is called transformation. Which is the classical tetracycline resistance gene? Come on. Depth. Tetracycline resistance gene. Any idea? Just mug it up. Or they will keep this and ask for which antibiotic. That time you must answer tetracycline. Except for mugging, there is no other choice. Tetracycline resistance gene. Single, any idea? Can you tell me a question for single? Single recognition site is best using restriction enzymes. If you know this point, you can answer a tough MCQ. So I have made it single. Ligation of alien DNA occurs in which site? Or they will ask, what is the speciality of restriction site? And they will give you 4 choices, which you cannot answer. So restriction site, you must read the question thoroughly. Question is important. Restriction. See, now you answer this. They can ask like this. E. coli is a selectable marker for what? E. coli. Antibiotic resistance. Now I think now you are little bit strong. Antibiotic resistance. E. coli, don't forget. Basic requirement of any ecosystem. They will give 4 choice. Water, sand, oxygen, sunlight. What is the basic requirement of any ecosystem? Same word in a textbook they are given. Be very specific to answer. Solar energy, sunlight. This is the basic. Primary production means what? What do you mean by the word primary production? Because secondary is different. Any idea? This is called the primary production. This is called the primary production. Primary production means this is called primary production. They will give this definition and they will ask primary production, secondary production, tertiary production, quaternary production. Be very careful while answering this. And this is called the primary production. What is the unit? What is the unit of primary production? What is the unit of primary production? Any idea? What is the unit of primary production? This is the unit of primary production. Can you define productivity? Productivity means the rate of biomass production. Or they will ask, the rate of biomass production is called 1, 2, 3, 4. 4 choice. Gone. The rate of biomass production refers to productivity. So what is the unit of productivity? 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 Unit of productivity. Dash can be utilized by plants during respiration. Dash can be utilized by plants during respiration is GPP. Dash can be utilized during respiration. It's called GPP. Organic matter rate of production. This is a dry topic, but you will have definitely 5 MCQs. Minimum 5 MCQs in this video. Yes. Ordinary organic matter rate of production. Any idea? Rate of matter of organic production during photosynthesis is called GPP. This is a separate MCQ. This definition itself is a separate MCQ. It's GPP. What is the question? Okay. Consumption biomass. What is the answer? Consumption biomass. Biomass. Available biomass for consumption is NPP. Is NPP. What is the question? Biomass consumption. What is the thing? Okay. These are all repeated MCQ points. What is this? Organic matter. That is biomass. This is organic matter. Rate of productivity. Is called GPP. This is organic matter. Rate of production, organic matter. Now organic matter consumers. Consumers organic, consumers non-organic. Now organic matter consumers. This is new, NEW, new. New organic matter consumers. Any idea? Rate of formation of organic matter is called secondary productivity. Don't forget the question. New organic matter consumer. Rate of production of new organic matter consumer is called secondary productivity. All the 4 MCQs which I discussed are very tough, confusing MCQs. Can you answer this question? Unit of productivity. Just now I discussed unit of productivity. Remember, see, we will forget things. That is how there is a proven method called 7 days continuous recall method. 720 out of 720 top students, they keep it as a secret. If you want to remember all these points in this video, you must hear this video for 7 days continuously. If you do it, you will remember all the 100 MCQs for 1 year. For 1 year. That is a proven method. If you don't practice 7 days continuous recall, tomorrow afternoon, you will not remember even a mustard. That's a yes. It's a proven method. See, just now I discussed, you forgot. Unit of productivity, gone. This is the unit of productivity. What is this? Unit of primary producer. You forgot. Tetracycline resistant gene. Gone. Gone. Tetracycline resistant gene. This is the purpose of 7 days continuous recall. Don't waste your time, okay? Very important. Name for antibiotics. Gone. You forgot. Caniamycin, tetracycline, chloramphenicol, and ampicillin. I made it very, very clear. Yes? Good. 70 billion tons dry weight. What is the question? 70 billion tons dry weight. 170. 170. Any idea? Same MCQ they have asked 3 years before. 3 years before the same MCQ they have asked. How to remember? Only 7 days recall technique. 55 billion tons. Any idea? 55 billion tons. Productivity of organic matter in ocean. Question is very, very important. You must know the question. Oceans. What is 70%? Ocean occupies 70% of Earth's surface. Change in species composition. Change in species composition in an area is called ecological succession. What is the definition? Change in is called ecological succession. See, whenever you see the word entire sequence of communities, okay. Whenever you see the word entire sequence of communities is dash. Entire. Whenever you see the word entire, think of seer. Yes. Or they will ask, what is the explanation for seers? And they will give 4 choices. It will be very, very tough MCQ. Seers. They can ask like that, seers. What is the answer? Seers. What is the answer? Individual transitional communities. Individual, whenever you say individual. individual. That time you must answer SERAL, seral community. Seral community. Individual, individual, seral community. Increase in total biomass occurs in individual, increase in total biomass occurs in successive seral stages. Or what is the benefit of successive seral stages? That time you must answer increase in the total biomass. This is how you should prepare for NEET. Not reading he found out that and I found out this and they found out that. You cannot remember even for half an hour. Succession occurs in areas which you have lost all the living organisms. All the living organisms, very important. What is it called? It is called all, A-L-L. Whenever you see the word all gone, you must remember it is called secondary succession. What is the definition? It is called secondary succession. Hisardale is a flower. Any idea? Hisardale is a flower. Any idea? Any idea? Hisardale. It is a sheep by cross breeding. Cross breeding. What is the question? Hisardale. DNA fragments attracts towards anode. Why? This is assertion reason MCQ. DNA fragments attract towards anode. Why? DNA fragments. Why? Because DNA is negatively charged. Because DNA is negatively charged. Seaweeds is used to make what? Seaweed. Any idea? Or they will ask, agarose is made from? They can ask like that. Agarose is made from seaweed. Agarose is made from seaweed. Agarose is a synthetic polymer. If you see a statement like this, what will be your answer? True or false? Agarose is a synthetic polymer. True or false? Because it is a natural polymer from seaweed. This is how you should prepare for NEET. Or they will ask, which of the following is a polymer or natural polymer? That time you must answer agarose. Size. Can you tell me a MCQ point to this? Which of the following decides the use of agarose? Size of the DNA, not the, size of the DNA determines the use of agarose. DNA stained with what? First you must know whether DNA can be stained. What is your answer? Can DNA be stained? DNA stained with what otherwise? Ethidium bromide. See, PYQs, previous year MCQs, they will not repeat at all. You must always prepare for new MCQs like this. Ethidium bromide. It is used for DNA staining. New MCQs. Can you tell me this? Successive serial stages. Just now we discussed. You forgot. That is I'm repeatedly telling 7 days continuous recall. Tell me the answer. You forgot. I made it very clear now. Serial community. Again you forgot. What is the answer? Yes. What is the question? I told you entire. What is this? Ecological section. What is the definition? See, you forgot. What is this? What is this? All means secondary. This is how you should prepare for NEET. Okay. See. Write some good comments in the comment section and share this video with your junior friends. Okay. And to your seniors and your classmates also. Okay. After hearing this video, you can hear my part 81 in the same YouTube channel. I have presented so excellently. Okay. So RNA is removed by proteases. True or false? RNA is removed by proteases. True or false? RNA is removed by ribonucleases. RNA is removed by ribonucleases. Purified DNA formation is dash reaction. Purified DNA formation is dash reaction. Or they will ask, which of the following is a precipitation reaction? That time you must answer bacterial cell is broken by chitinase. Chitinase. Bacterial cell is broken by lysosome. Or they'll ask, what is the function of lysosome? 4 choices. Gone. Lysosome. Bacterial cell wall breakage. Fungal cell is broken by cellulose. Fungal cell is broken by cellulose. Fungal cell is broken by chitinase. Fungal cell, chitinase, broken. Plant cell is broken by cellulose. Plant cell is broken by cellulose. It is a false statement. Can you agree with me? Plant cell is broken by cellulose. It is a false statement. Can you agree with me? Yes, it is a false statement because you didn't see the spelling correctly. Cellulase. This is how NEET people will confuse you. What is the question? Cellulose. What is the answer? Cellulase. A. Be careful. Okay. Can you tell me the definition for incubation? Incubation. Any idea? Incubation. Example. Restriction enzyme digestion is done by incubating purified DNA. Purified DNA. What's the question? Incubation. Incubation. Insertion inactivation. Insertion inactivation is related to tumeficans. True or false? TUMIFACIANS. Get prepared for new MCQs, not old PYQs. They will not repeat the MCQs at all. The correct answer is beta-galactosidase. Beta-galactosidase is responsible for insertional inactivation. So, insertional inactivation is by beta-galactosidase. Pathogen of dicot plant. Pathogen of dicot plants. A dangerous pathogen for dicot plants. Any idea? Any idea? Agrobacterium tumeficans. It is a pathogen of dicot plants. How to remember this? Only one method, 7 days. You will be the master. Pathogen of dicot plants. Have you heard this word capital T DNA? Capital T. Not small t RNA. Capital T. What is the answer? Capital T DNA. Any idea? Tumor in plant cells. TUMOR. Tumor means cancer in plant cells related to agrofumicants. What is the question? Capital T DNA is related to tumor in plant cells, agro. Kahn's technique is started in which place? Kahn's technique. Do you know Kahn's technique? Here my previous video I given so nicely. Kahn's technique was started in Bangalore. What is niche specialization for? Niche specialization has greater species diversity. They will give low. Be very confused. Be very careful. Greater species diversity is typical for niche specialization. naturalistic and geographer. Who is a naturalistic plus geo, geographer? Any idea? Alexander von Humboldt. He is a naturalist and geographer. Naturalist and geographer. Naturalist and geographer. Rectangular hyperbola. Have you heard of this? Rectangular hyperbola. It is a relationship between species richness and area. Species richness and area. Species richness and area. Hyperbolic. What is this definition meant for? Log S is equal to, as per your MCQ, as per your textbook NCRT, log S is equal to log C plus Z log A. How to remember? 7 days recall. Log S. Can you expand everything in this? Can you expand everything in this? What is S? What is S? What is C? What is Z and what is A? Any idea? S for species richness, A for area, Z for regression coefficient, C is equal to y minus intercept. C is equal to y minus intercept. Always remember regression coefficient. They have asked us MCQ once. Regression coefficient. Yes. Fruit eating birds are called what? Fruit eating birds are called frugivores. Frugivores. Fruit eating birds are called frugivores. David Tillman. What did he found? David Tillman. Species diversity of ecosystem. David Tillman. Species diversity of ecosystem. Increased diversity gives increased productivity. True or false? Okay, you must tell this. Increased diversity gives increased productivity. True or false? It is a It is a true statement. So increased diversity, there will be always increased productivity. It is a true statement. Paul Ehrlich. What is related to what? Paul Ehrlich. He is related to RIVET, Popper hypothesis. I think it's something related to aeroplane. That rivet, RIVET, related to aeroplane. Some aeroplane hypothesis. It's given in a textbook only, something related to aeroplane. AEROPLANE. So keep it in mind. Whenever you see this MCQ, think of aeroplane also, okay, in solving MCQs. Revert popular, the first answer is Paul Ehrlich. Maybe he would have gone in an aeroplane. Calcium decreases. This is an MCQ. Come on. Calcium decreases, very important, okay. The efficiency of DNA entering into a cell. What is the correct answer? Very important MCQ point. See, PYQ, they will not repeat even a single comma. Always learn new MCQs. What is your answer? Calcium increases DNA fastly into the cell. Calcium increases. Calcium increases. The word tumor in plant is related to, just now I discussed tumor in plant. I am talking about plant. PLANT, very important, plant, related to. Tomorrow afternoon you will forget. Yes. Before that only you will forget. 7 days continuous recall in plant. Cancer causing virus in animal. I am talking about animals. Some relationship in your textbook is given. I think now they have found out all new techniques. Retrovirus in animals. Retrovirus in animals. Dash is used to study restriction, enzyme, digestion. See, when they give 4 choices, you cannot answer. Study very thoroughly. The answer is agarose gel electrophoresis. Agarose gel electrophoresis. What is the question? Agarose gel electrophoresis. Dash inhibits the decomposition of detritus. I am talking about inhibit. Read the question very well. Inhibit. Low temperature and anaerobiasis. Don't forget the word inhibit. Inhibit. Low temperature and anaerobiasis. Taylor experiment. Taylor experiment. He proved DNA. He proved DNA in a chromosome replicate semi-conservatively. Semi-conservative. What is the Taylor experiment? He proved replicate semi-conservatively Taylor. E. coli divides in Taylor experiment. True or false? This is the depth of MCQ. Depth. E. coli divides in Taylor experiment. No. E. coli divides in this is the actual NEET preparation. Have you heard this? Do you know this experiment? If you read this experiment, you will know. E. coli. E. coli. What will happen in this experiment? E. coli divides. Hybrid DNA formed in generation 1. Very tough MCQ, okay? You have to think and answer. Hybrid DNA formed in generation 1 of Michelson experiment. I am talking about hybrid DNA. And I am talking about generation 1. And I am talking about Michelson experiment. 3 words are important. Are they true first? Is it this experiment correct? Think. Hybrid DNA, generation 1, Michelson experiment. It is a true statement. There is a diagram in your textbook. If you memorize the diagram, you have to mug it up. You can answer this. Memorize the diagram today itself, okay? A light DNA is written as how? How is a light DNA written? Fourteen N. 14N. This is light DNA. Both are 14. Light DNA. How is a hybrid DNA written? Hybrid DNA. 14, 15. Hybrid DNA. How is a heavy DNA written? Nobody will teach you like this. Only Sheben can do this. Heavy DNA. 15N. 15N. There is heavy DNA. Light DNA is formed after 20 minutes in Michelson's experiment. True or false? After 20 minutes, light DNA. Light DNA is formed after 40 minutes. These are all tough MCQs. Only people you are lucky enough to hear my video. Yes, you are lucky enough to hear my video. Share my video with your good friends, okay? Three million base pairs found in human genome. True or false? 3 million base pairs found in human genome. True or false? It is 3 billion. 3 billion. Repetitive DNA is studied by repetitive DNA is studied by repetitive DNA is studied by DNA fingerprinting. Don't forget Sheben, okay? Micro is a type of repetitive DNA. Micro, MICRO, is a type of repetitive DNA. Micro is a type of satellite DNA. Micro, MICRO, is a type of satellite DNA. Micro. High degree of polymorphism is the basis of what experiment? High degree of polymorphism is the basis of what? It's the basis of fingerprinting. Or they will ask, what is the main basis for DNA fingerprinting? That time you must answer high degree of polymorphism. Both sides repeated MCQs. Independent in idea. Independent in idea. Plasmid replication within a bacterial cell is not depend upon the chromosomal DNA. It is independent. It is independent. In 2004, in India, they prepared the IUCN Red List. IUCN Red List in 2004. 784 species in India. 784 species. IUCN Red List mentions extinction of 784 species. 338 species in India. Out of 784, 338 are invertebrates. Out of 784, 388 are invertebrates. What is 359? Are invertebrates. Invertebrates. 338 vertebrates. 338 is vertebrates. 359 is invertebrates. 359. Okay, good. 87. Out of 784, 87 are plants. What is 27? The last 20 years has witnessed 27 extinction. How to remember all these MCQs? 7 days continuous recall. Recall technique is very important. Revision is a useless waste of time. You must read the textbook once. Then you must come to this video. Keep on recalling. Recalling. It will go depth. Birds are the highest risk of extinction in India. Birds are the highest risk of extinction. True or false? Highest. Highest is always amphibians. Is it not a tough MCQ? How to remember this? There is only one method. Recall. Recall. Order of percent of extinction. Order. So, see, I have given 5 MCQs here. They can ask in this descending order, shuffling. One tough MCQ. Or they will ask percentage for an individual thing. So, totally 5 MCQs are in front of you. What is the question? Order of percent of extinction. Disappearance. Extinction. So, how much? 32 amphibians, 31 gymnosperm, 23 mammals, 12 birds. 4 MCQs clean for you. Or they will ask descending order or ascending order. So highest is for amphibians, lowest is for birds. You can ask, is it not a rank deciding MCQ? Is it not a rank deciding MCQ? $15,000 species, 500 species. What is the question? Risk of extinction worldwide. Worldwide risk, not extinction. Already they are in the risk. 15,000 species. What is over exploitation? All repeat MCQ. How much? What is the risk? What is this? Over exploitation. Any idea? It is a cause of biodiversity loss. It's the same word is given in your textbook. Same word. So don't keep on reading PYQs. They will not repeat any PYQs. But you must know, you should apply, do PYQs to have an idea. To have an idea, that's all. They will not repeat any PYQ. You must always, every year you will come with new MCQs. You see, this is a very important MCQ. Over exploitation. So, stay tuned with Dr. Shiban. Share this video with your friends, okay? Stay tuned.